Yeah,
Cool.
I can see it.
Cool. Thank you. All right. We'll wait a few more minutes and then we'll go ahead and get started. All right. We'll start in another like 2-ish minutes. Okay. I'll go ahead and get started. Hi everyone. My name is Pranay. I just graduated from Duke in BME this past year. And I'm gonna be teaching this, I guess, like class slash fellowship for you guys. And everyone can see my screen all right when I present it, right? Actually, I'm just not gonna present it so I can see your like reactions and stuff. Can you see like this slide though that I'm on?
Yes.
All right, cool. Thank you. Okay, cool. So yeah, my name is Pranay. Um, this is just like a short introduction before I guess we do a quick icebreaker and then I'll get into the content. But this is a little bit about me. I'm from Northern Virginia. Uh, did high school at a magnet program called the Academy of Science, which introduced me to, I guess, the field of research and I guess got my interest in like science and engineering up quite a bit. For the past 4 years, I've been at Duke, majoring in BME, and doing a lot of research with professors, taking super interesting classes. Really love that experience as well. And then starting next year for the next few years, I'm going to be at the University of Pennsylvania, um, doing a PhD in bioengineering. Um, so I guess that is my like education journey so far. And then at any time, if you guys have any questions about like my experiences or anything, even once we start the content, just feel free to like unmute and jump in. I want this to be like actually useful for you guys. So if there's something I'm talking about where you want to know more about it or anything, let me know. Also, if there's something where, I might be going too in depth and you guys don't care as much about it, also let me know. That's super chill. Um, so this is a slide with some of like my background, I guess like work, rather than education. So when I first got to Duke, I joined a lab that did a lot of immunology work. So basically trying to develop therapies to essentially train our immune system to recognize foreign things and take action against it. So training our immune system to, for example, be able to fight COVID better or fight the flu better. So that's the research I did there for my first 2 years of college. I wanted to explore another side, I guess, of biotech and of biomedical engineering. So I interned for a company called Medtronic for 2 summers. Medtronic makes a lot of different medical devices, so pacemakers, devices for like cardiac surgery, all sorts of different stuff like that. So I was there for 2 summers, enjoyed that experience, but kind of realized that the more like molecular and cellular engineering side of things was more interesting to me personally as opposed to like medical devices. I then did research with a group at Duke called the Chatterjee Lab, or like the Programmable Biology Group, where basically we tried to apply machine learning and AI to different problems in biology and basically develop algorithms to try to develop new therapeutics, new proteins, and things of that nature. And then most recently, I've been doing work with a startup where our goal is basically to try to predict the success and efficiency of different drugs in model cells rather than in humans. And so this, I guess, is my primary area of interest. It's the intersection between machine learning and synthetic biology. So basically, how can we use machine learning and different computational approaches to predict how biology will act, essentially, and how can we use that to design new biology? So I'm gonna go ahead and start like I guess the content for this week in a few minutes. Before then, since we don't have too many people on the call, I'd love it if we could just do like a quick icebreaker and I could just get like a name, your grade, and like one thing you're interested in. If that's fine with you guys, I'll just like call you guys out, and then if you are chill with it, just unmute yourself and I guess just answer those 3 things. I'll just go in order in the order I see them on my screen. So I guess first is an A.
Oh, hi, my name is Anay, and I'm going to ninth grade next year, and my interest is also like kind of the intersection of mechanical learning and like machine learning kind of and biology, like kind of combination of the.
Cool. Nalby or Nalby.
Hi, my name is Nalby. I am going into my junior year, and I'm also really interested in like the applications of machine learning into biology fields as well.
Cool. Anjan. Oh, never mind. Adya.
Hi, I'm Adia. I'm going into my sophomore year, and I'm interested in something related to neuroscience and ML.
Okay, cool. Jocelyn.
Hello, my name is Jocelyn. I'm also an incoming sophomore, and I'm interested in like computational neuroscience.
Cool. Tanvi.
Hello, my name's Tanvi. I'm also going to go to my sophomore year, and I'm interested in biology and neuroscience.
Cool. Netra or Netra.
Hi, I'm Natra. I'm a rising sophomore, and I'm mostly interested in cancer research.
Cool. Nikita.
Hi, my name is Nikita, and I'm going into my sophomore year, and I'm interested in molecular cell development.
Cool. Raghav.
My name is Raghav. I'm a rising junior, and I'm going into like I have an interest in healthcare and biology.
Cool. Pranav.
Hi, I'm a rising sophomore and I'm interested in applying CS concepts into biology.
Okay. Anika.
Hi, my name's Annika. I'm a rising senior and I'm interested in neuroscience.
Cool. And Shreya.
Hi, I'm Shreya. I'm a rising junior and I'm interested in biology, but I guess more specifically genetics and reproductive health.
Okay, cool. Um, okay, nice to meet you all. I guess this will be like the cohort for our classes and stuff. So again, just like anything you're interested in, anything you want to stop me about, even if it's not on the slide, if it's just about my experience or like anything, I guess, just stop me. Just unmute and just say whatever you want whenever you want. I want this to be like pretty chill, so definitely don't hesitate to speak or anything. Okay. So as far as I understand, you guys are doing like I guess like a course, and so for biotech. And so I guess this week's presentation is just kind of like an introduction into what biotech is, different things that are happening in the field, um, and maybe I guess just like introducing you guys to the field so that you guys can start thinking of ideas yourselves and for different projects that you might want to take a look at. Okay, so basically, what is biotech? It's basically all about using living things, whether that's bacteria, cells, DNA, to create useful products or to solve problems. And the range of these problems is honestly huge. It can involve things like medical devices, as I mentioned, if you're interested in like making pacemakers or if you've heard of, for example, Elon Musk's company Neuralink, trying to modulate brain signals, whether it's more like hardware focused like that, or whether it's actually developing medicines, creating genetically engineered crops, for example, to be more drought resistant or pest resistant, or even in like environmental engineering, like cleaning up pollution, using different sort of engineered microbes and things like that. So basically, it's like combining biology with technology and engineering. And so I think we're gonna in this like course, we're making it a pretty big point to try to expose you guys to like the toolkit of different possible things in biotech. Because as far as I've seen, at least through my undergrad in BME at Duke, we do a lot of work to try to look at the different tools in biotech, and those tools can really be applied to all sorts of different problems in the field. And so I guess just one example, or I'm going to run through a few examples of different things that would surprisingly or unsurprisingly fall into, I guess, the area of biotech. One example of that can be as simple as like brewing beer with yeast. So basically, taking advantage of the fact that yeast, a certain type of fungus, can break down sugar and turn it into alcohol and carbon dioxide through fermentation. So we take a known pathway in biology and basically use it for something that we want, which is in this case beer. So this kind of like flow chart or like the flow of information is super, super common in biotech. We take something that we know or something that we hypothesize. For example, if I have a specific pathway that I know the human body naturally uses to kill cells. And then it's all about taking what we know and applying it to a new problem. So let's say I want to take that pathway and now engineer different portions of it to instead hijack that pathway and use it to kill cancer cells. That's something that's super doable, and super interesting to look at through the lens of biotech, which we will go through. Another few examples, I guess, of different things that would fall under biotech are technologies like CRISPR or mRNA vaccines. So if I don't know how familiar you guys might be with CRISPR, but it basically stands for clustered regularly interspaced palindromic repeats. So basically, it's a system that we can use nowadays for gene editing. So let's say there's a specific gene that's present in someone's body that we know leads to a specific disease. We want to go in and we want to, let's say, delete that gene. That's something that's really possible with CRISPR. CRISPR basically acts like a pair of scissors, goes in and cuts that gene out. We can then either let the body repair it itself or actually introduce a new gene into the space that we cut the old one out of. And so recently, I think as of a few years ago, or maybe in the last year or two, there have actually been therapies that have been launched using CRISPR technology. The first one that was FDA approved was for sickle cell anemia. So they approved a CRISPR-based therapy where they can actually go in and edit the gene responsible for causing sickle cell anemia, which is super, super interesting. Technologies like this, there's also a lot of research into the ethics behind it. There's another example where a few years ago, a researcher in China used CRISPR or kind of like bypassed all of the regulations around CRISPR and basically used it to engineer twin girls' DNA in the embryo itself. And so this was a huge thing in science. It did work, which was a huge thing, but also it just showed how easy this kind of stuff is to do nowadays, which is also a little bit scary. And then you also have things like mRNA vaccines. If you've gotten like the COVID vaccine, I think I like any of the major ones, you've gotten an mRNA vaccine before. And so I guess the most recent example of this, like I just mentioned, was COVID. So basically the idea behind mRNA vaccines is that if you've taken like an intro bio course, you've heard that DNA makes RNA and RNA makes protein. So the idea behind this is that we know that there's specific viruses like CRISPR, which have certain proteins surrounding them or on them. Um, and so I think this, sorry, I think this specific picture is showing it for for the flu, sorry. So it has a spike protein, let's say. So basically the idea is, can we deliver the RNA which codes for that protein and actually train our body and train our immune system to actually be able to recognize it and then destroy it by making its own antibodies against it. So if you're interested in maybe the field of immune engineering, that might include trying to engineer different pathways that our immune system uses to recognize and destroy new foreign cells, foreign diseases, and things of that nature. And so the big picture of these different technologies, and the reason I'm telling you all of this, is that these things are really, really cool, and they are active technologies that are being used currently to change medicine, to develop therapeutics, and to change people's lives. And so even if you think about a simple use case like making beer, all the way to this, that's basically all in the field of biotech and in the realm of biotech. And these are all high impact things that the field of biotech allows people to do. And so I've kind of touched on a little bit of this indirectly, but I think it's important to like actually talk about the exact reasons why biotech matters. And so again, it's a really, really huge field. So it's important for a lot of different things for in terms of affecting people's health and helping people's health. It can help us treat diseases faster, treat diseases better, detect diseases faster, before symptoms appear. It can help us diagnose different diseases very rapidly. If you think of our COVID tests, like PCR tests, those are all developments that happen in the field of biotech and bioengineering, and allow us to do a lot there. If you think of how people actually get diagnosed with cancer, a lot of times it's a biopsy. So a lot of these medical concepts and medical tools that people nowadays use to diagnose different diseases and identify them, all came through developments in biotech. You can also think of different therapeutics, which might be the biggest area in biotech that we're most aware of, and one of the most quick moving areas in biotech. So basically, this can include things like biologics. So whether it's like an antibody treatment, and stop me if you guys are like completely lost, but when I say something like antibodies, I'm gonna guess that we sort of know what those are, but I guess I'll do like a brief introduction. So there's typically in the example of like trying to treat a disease, you have something called an antigen, which is like a bad particle. Let's think of it like that. And then your body or your immune system is going to use an antibody to bind to the bad particle and degrade it in most cases. So there's a lot of work being done on engineering those antigens to be better at binding bad things or being better at binding specific things. So, for example, if you know that there's a specific cancer that has a specific antigen or bad particle associated with it, you can think of the task of maybe trying to design a new antibody to bind to that better. So there's a lot of work in that kind of area in the field of like therapeutic development. You can also, for example, look at why biotech might matter in the area of food. You see things like improvement of crops, meaning making them more drought resistant, or more, um, sorry, or more like insect resistant. And so that can reduce the amount of pesticides we have to use that can help protect different animals, things of that nature. And so these are all things that might fall into, for example, like genetic engineering or gene engineering. You hear people talking about like GMO foods. While that might sometimes be bad, GMO foods are really good. They help us optimize like the food growing process. They make our food healthier to consume. We now need to use less pesticides when we're making the same food because now we can engineer the actual plants themselves to be resistant to pests. You can think of things like environmental engineering, which also falls under bioengineering or biotech. This is, for example, like something like carbon capture. If you think of a big factory which releases all that pollution, can we actually engineer like a bacteria to be able to take that smoke and convert it to something useful. That's another application. And so the idea here is that there's a lot that falls under biotech, and all of it matters a lot. So I touched on these a little bit, but yeah, diagnostics, therapeutics, digital health, I guess I didn't really touch on as much, but I think I will just because a lot of you seem to be interested in the intersection between like AI plus biology or plus healthcare. Nowadays, there's a lot of different products and a lot of different tools in the area of digital health that make it really, really interesting or lucrative to learn about and to go into as a career, to develop new products in. And so whether you think of wearable devices like an Apple Watch, like the Whoop band, the Aura Ring, things that track your sleep, things that track your habits, things like that, these all fall under digital health. You also have AI now being used to discover new drugs. So that's kind of along the lines of the research that I work on, is basically how can we learn things about existing drugs and existing proteins to maybe extrapolate that to new ones that can be used for new diseases or new indications and things like that. And so again, like whether it's an Apple Watch, which now can track different things like arrhythmia or like odd or wrong rhythms in your heart, whether it's hospitals now using AI to help identify things that are wrong with X-rays and different things in radiology, or whether it's the different companies that are now being developed to try to address mental health issues by making AI chatbots and things like that. These all fall under, the umbrella of biotech, and I guess more specifically of digital health within biotech. Okay. So I guess for today, we're gonna do a little bit more of a deep dive into diagnostics and therapeutics. I think next week we're gonna go more into digital health and the sustainability side of things. Okay. And then actually, wait, I'll pause here to see if anyone has any questions about anything I've said so far. If not, super chill, and we can just keep going. But if anyone has any questions or anything.
I had a question.
Yeah, what's up?
So, I think you mentioned stuff like CRISPR and gene editing, but
Yeah.
how would we get access to like those materials? Like, how do we actually um do research on that type of topic when we don't have access to our own labs?
Yeah, okay. So that's a really good question. And so the thing about CRISPR and honestly anything in biotech is you can plan it out pretty well before you actually have to like test it experimentally. And so what that means, for example, let's say that you want to look at sickle cell anemia, which I said is one of the diseases that CRISPR now can treat. So let's say you want to do a project where you want to investigate sickle cell anemia. One important thing that you might want to do if you want to treat it with CRISPR is try to see what specific gene in the genome might be responsible for sickle cell anemia. So that might be doing a little bit of bioinformatics work to see what genes are present in patients with sickle cell anemia versus without sickle cell anemia. And so once you identify those genes, you can take a look at, so for CRISPRs, there's different CRISPRs that basically can recognize different sequences of DNA. So not all CRISPRs are the same. So once you find the gene that might be common among sickle cell anemia patients, you can go then try to find a CRISPR that, would match that gene or that might be best for that gene. There's also nowadays algorithms that are coming out to try to predict how one biomolecule might interact with another biomolecule. So then once you have your gene sequence and your CRISPR sequence, you can use one of those algorithms to try to see, oh, does this algorithm actually think that it'll bind to it with high affinity? Affinity meaning like how tightly that binding interaction will occur. And so basically, I guess like long story short, that was kind of a windy way to answer it, but there's a lot of work that you can do ahead of time in terms of planning, in terms of research. That you don't necessarily need to have access to like a lab to actually do. Obviously, testing it in a lab will actually tell you whether it works or not. but I think you can actually get a lot of progress towards a specific interesting project without necessarily having a lab. I don't know if that answers your question a little bit. Of
Yeah, it does. Thank you.
course. Any more questions?
Yeah, I have a question.
Yeah.
You mentioned that in digital health there are a bunch of products and tools. So like could you like specify which ones and where you can get them?
Yeah. So I guess here, okay, let's see. Okay, so I guess like within digital health, again, it's a broad area, but if you think about something specific like AI plus biology, right? So let's say that what you're interested in is being able to design protein therapeutics to bind to like problematic ones, let's say. You can take a look at something like AlphaFold. You can take a look at something called Rosetta Fold, RF diffusion. These are all algorithms that have come out relatively recently that all help towards that task. If you want to do a project more so like, let's say, digital health in terms of like a mental health resource, let's say is what you're interested in. Um, I don't know any specific companies that are doing that off the top of my head, but that would be a project where more than more than likely you'd be doing some sort of fine tuning on an existing language model. So for that kind of project, I would maybe take a look at um, like prompt engineering GPT and using like OpenAI's they're called APIs. You can look that up. Basically, in order to use GPT for a specific use case. And then if you want to take a look at, let's say, like an oxygen sensor or like a glucose sensor or something like that, if you're interested in more like the fitness or sorry, the wearable or like hardware side of things, the way that I would maybe think about doing that or exploring something in that is you can order like an Arduino, which is basically like a little computer that you can like engineer and like code up yourself. You can buy different sensors for that. And those are all ways that I would go about doing a project like that. I don't know if that kind of answers your question.
Yeah, it does. But what specifically could you do for like neuroscience?
So for neuroscience, I think that one thing that might be interesting there is so I know there's a lot of data on like brain scans or like CT scans, EKG data. Um, so I think that there's a lot of like these are data sets that you can probably pretty easily find. You can see different like for example, a project that I worked on a few years ago was we had a data set of like basically pictures of spirals that different people drew. And basically there was a data set of spirals that, healthy individuals drew versus spirals that like patients with Parkinson's and Alzheimer's drew. So a project that I worked on a few years ago was, can I develop a machine learning model to basically take in an image and try to predict whether the person who drew that spiral is healthy, Alzheimer's, Parkinson's. So that is an example of a project. So I would think that for neuroscience specifically, I would start thinking about what kind of data might be available to answer the kinds of questions you want to answer. So let's say that you are interested in brain cancer. I would maybe take a look at CT scans to do some sort of like tumor identification or something like that. So I would really be looking at like the intersection between like specifically in machine learning, I think that looking at some sort of like image identification or image processing would be pretty interesting to look into there. I don't know if that answers it a little better.
Yeah, so another one. So how do you access that data?
Most of this data is like very much open source. So you can look up just something like CT scan data for X disease or something like that. And I have no doubt that you'll find at least something. Um, Either on Google or a lot of times ChatGPT also, if you ask it for data sets, will give you the link to different data sets you can access. So those shouldn't be too hard to find. Most of these are like not behind paywalls or anything. It's like a lot of data is available for free.
Okay, thank you.
Of course. Any more questions?
I had another one.
Mhm.
How do you go about the machine learning? Like you mentioned, using a machine like an AI to detect like from a person's drawing if they have Alzheimer's.
Right.
How do you create your own AI and train it?
Okay, I mean, so there's a lot of different like machine learning algorithms that you can go about trying to use. There's um here, like there's I think you guys can see my screen, but like for example, I would just go about like trying to use different like free online resources to like help do what you want to do. So let's say you're interested in image. Basically, what that problem is image classification using machine learning. And so this is an example of a tutorial you could watch and follow. Okay, I don't really want to watch an ad right now, but a tutorial you could watch and follow. Basically, what most of these projects would involve, if you're interested, if any of you are interested in, I guess, machine learning or AI, would be in Python, which is a programming language. If you don't have experience using Python, um, that I think that that is definitely a skill that you should try to learn or pick up if you're interested in this kind of, area. And that's also something that's super, super, super easy to follow along or learn just through like YouTube or something like that. So if that's something you're interested in, just like follow YouTube tutorials, and just and like familiarize yourself with it. And I think it's very, very doable. So I guess to answer your question, like how would you implement it? How would you go about it? Basically just look up how other people solved similar problems using Python or like using a different programming language.
Okay, thank you.
Yeah.
On
Also,
that note, do you recommend us like using a previously available model, or like maybe training a new one using like TensorFlow or like PyTorch or something like that?
I think that for the sake of learning, which I think that that should be the goal at this stage, especially like in high school, you want to like learn and build things yourself. Obviously, like using someone else's like pre-made model and just applying it to a new problem could be worthwhile or could be useful. But I think there is more benefit in you learning to develop and train your own model. Even if it's bad, like that is an impressive project for a high schooler, nonetheless, making and training your own model. So, yeah, I think that I would recommend trying to train your own rather than using a pre-trained one. And as far as like PyTorch versus like TensorFlow or whatever, um, it doesn't matter which one you use. Both will allow you to have the ability to make and train your own models. I personally like TensorFlow for easier or smaller projects, PyTorch for larger ones. For you guys, for the people that are not familiar with these are basically like libraries within Python that basically make doing machine learning super, super easy. So once you get a hang of like Python basics, I would recommend maybe watching a tutorial on like PyTorch or TensorFlow, which basically make machine learning in Python easier. So I guess Ane, like what I would recommend is probably try to make your own. And it doesn't matter which one you use in terms of like TensorFlow versus PyTorch.
And the accuracy, like if it's even like a little bit like worse than somebody else's, you still recommend like, you making my own?
I mean, it depends like what your goal is, right? Like if your goal is to make yeah, it depends what your goal is. If your goal is to like make the best possible model, um, then if it's worse than someone else's, then maybe use someone else's. But if your goal is to, learn and experiment with different types of models, get experience in, making new ones, then I would maybe recommend making your own. But yeah, it just depends what your goal is there.
All right, thank you.
Yeah. Any more questions? Okay, cool. We will go back to this then. Where was I? Okay, so yeah, for today, we are just gonna go into like diagnostics and therapeutics. So I think this goes until 8:30, but I want to leave at least the last 30-ish minutes for a discussion thing that we have at the end of this. So I might speed up a little bit or skip over some things that I think aren't as interesting just based on what you guys told me you guys are interested in. But yeah, today's topic, diagnostics and therapeutics. Diagnostics, obviously, as you guys know, like help detect or diagnose a different a biological condition, whether it's like an infection, whether it's like a mutation in your genes that leads to a disease, whether it's a specific hormone level, or whether, let's say, for example, like diabetics, it's really important for them to know their blood sugar. How can we go about identifying what the level is and then actually taking actions based on that? So there's some key qualities of diagnostics that are super important to think about. You want ideally diagnostics to be fast, cheap, and accurate. Accuracy comes at the top priority. Fast and cheap, I think, are the second most important, I guess, like bucket of categories. But when you're making a diagnostic, you want it to be accurate. And even if it's like a physical device diagnostic or an algorithm or like a computational biology type project that you want to be used in a diagnostic setting. I don't remember who was asking about it, but for example, let's say that you want to try to diagnose cancer through X-rays. The key thing that you want to be looking at there is some level of accuracy, right? You want it to be better than a random guess, or you want it to be better than the next possible, or the next best way of doing it. So these kinds of diagnostics are actually super interesting. Because they can be used in all sorts of different environments in low resource settings, different countries where they may not have access to different, medical resources. When you think of a pandemic, we're really prioritizing speed there because we need to know whether or not someone has COVID or doesn't have COVID. And then also for things like preventative care, like for the example of diabetes that I mentioned. So this is a slide which basically shows how CRISPR can be used as a diagnostic tool, and these are two different platforms that have been developed to do this. The details behind this, I think, are pretty complicated and not necessarily needed for me to go through here, but the point is that you can take existing technology and apply it to new problems. So even though I told you that CRISPR was for, you know, cutting DNA and gene editing, um, the part that I kind of left out was that before it actually does the cutting, it has to, and I kind of touched on this, but it has to identify a specific region of DNA, bind to it, and then do the cutting. So we can kind of leverage the fact that first CRISPR has to bind to DNA to actually use it for the purpose of a therapeutic, or sorry, for a diagnostic. So let's say we're trying to see if a piece of DNA, let's say DNA X, is there or not there. We can use a CRISPR to try to see if it's there or not there by seeing whether or not it binds to it or doesn't bind to it, which is basically like what this is showing. You can also look at things like biosensors, which again would be something like glucose, or if you've been in like a chemistry lab in high school, you might have seen like a pH sensor, for example. Let's say that you want to measure the amount of protein that's present in a sample. These are all different, I guess, things that would fall under the umbrella of biosensors, which is a big tool that we might need. And the reason that this might be useful to you guys, even though a lot of you are doing, like computational biology or machine learning focused projects, probably, is the data that you use and that you collect for your project is going to be super, super important. And so you want to make sure your data is of the highest quality and is actually data that'll help you make the kinds of conclusions and make the kinds of, models or whatever that you might want to make. And so when you're collecting your data and looking at your data, it's super important to think about where that data came from. And so what sensors were used to collect this data? What patients were these sensors used on to collect this data? That's all really important stuff that you definitely should be thinking about when you're doing your projects and when you're going forward. Another tool that kind of falls under this like diagnostics umbrella is microfluidics, which is basically the science of manipulating really small amounts of fluids, hence the name microfluidics, also called labs on a chip, if you've ever heard of that terminology. So instead of running huge, big, full lab tests using big beakers, pipettes, things like that, microfluidics as a tool lets you basically perform chemical reactions, mix fluids, analyze samples, all that kind of stuff, just by using a tiny, tiny amount of a liquid. And so basically, it's a tiny chip, like microscopically sized, that has tiny channels etched into it. And so you can see those like lines here, for example. And so that these lines can control how different fluids interact with one another, how they move, how they mix, things like that. So this basically scales the process of science down to a tiny, tiny chip and allows us to do things fast, portably, and low cost. And so if you think about different diagnostic tools that may, like use your saliva. If you go to like a doctor's office and you need to do like a saliva test, or like for example, if you think of a company like 23andMe where they collected saliva to do DNA tests, they're not really doing things at a huge, large volume. They're doing things in a tiny quantity. You take a vial, you spit in it, and you send it there, and they do like gene sequencing on this. So they're using tiny amounts of liquids to do really important work. And so you can combine tools like microfluidics with tools like biosensors to create really, really powerful diagnostics. Um, or I guess I really did touch on this a lot, but main point here is during COVID is where we saw a lot of these diagnostics, like tools and techniques coming up quite a bit. We saw things like at-home PCR tests really, really emerge. And so we now have a lot of ways to test things really efficiently at home with these kinds of different tools that we have available. Another really cool example, which should, I think, in theory, be of interest to some of you, is like smartphone-based eye exams. Um, there's a lot of different eye-related diseases, or even beyond eyes, let's say like heart-related diseases that can be scanned via X-rays or via scans of some way. And so trying to diagnose these kinds of diseases or problems just by like pictures or scans is a really, really interesting problem in the field, and is something that where a good solution to this problem would really help a lot of people and also be a very, interesting and lucrative business idea as well. And so there are companies and people out there where they are doing things like trying to use your own phone, and basically make modifications or whatever, introducing a new camera or like a magnifying glass on top of your phone camera to basically collect eye scan data, and then basically feed that through some sort of algorithm to try to identify different diseases or different conditions that may be present based on the scans. These kinds of things are super, super helpful or important in like rural or low resource areas where they might not have access to like the huge machines that, different hospitals might have access to. These kinds of things also enable the super early detection of very treatable conditions, which can help prevent things like avoidable vision loss, or like avoidable heart attacks, or like tumor development or whatever down the line. And so if you think about why this kind of thing falls into the field of biotech, it combines physics and engineering through optics and how we're actually collecting that data. It combines diagnostics, which is our goal by, predicting whether or not a disease is there or not. It also combines digital health, like incorporating all of this and wrapping it up into like a smartphone app to basically tell you the findings of the experiment. So again, some use cases for diagnostics, low resource settings, super important there. Emergency rooms, when you're thinking about the speed of doing these types of experiments and doing these kinds of diagnostics, you want some sort of really, really fast results in an emergency setting. You also think about things like at-home testing, where you actually want privacy and want it to be super easy for an everyday user to be able to conduct. That's another example where these kinds of diagnostics would be super helpful. So yeah, when you're thinking about like what kind of project you might want to do, one thing, if you're interested in the field of diagnostics or like using machine learning or AI for identifying or diagnosing some sort of disease, you might want to think about like, how can we do X for cheaper than it currently is being done? So whether that's like diagnosing some sort of cancer from an X-ray. You think about how that's currently being done. A person needs to go into an office, they need to get an X-ray, but then even after they get an X-ray, they need to wait for a second appointment where a radiologist looks at the X-ray in front of them, analyzes it. That whole process could take weeks. So some of the pain points there are it's expensive to get the X-ray, and it's really time consuming and also even more expensive to have someone actually review that X-ray. So let's say that I want to go ahead and try to make something to solve this problem. Um, let's say that what I want to address there is the time between when you get your X-ray and between when someone like a radiologist looks at it and analyzes it for you. So I might then use X-ray data to then try to predict whether a cancer is there or not, so that we can try to, once they get the X-ray, move things along quicker. So how can we make a diagnostic for cheaper, better, more accurate, more efficient than it's currently being done or used? I'll pause there or I'll pause there real quick to see if anyone has any questions about like diagnostics or like that area in general. Um, if not, I'll move on. But any questions are totally okay.
Would like a classifying like algorithm, like would like that kind of diagnostic tools?
Yeah, like for example, when I said earlier, like using spirals or using whatever sort of data to identify between like healthy disease condition A and disease condition B, that can be done with a classification model, basically classifying a given data point into either category A, B, or C. So that's an example of like that is a diagnostic algorithm or can be used for a diagnostic purpose.
Okay.
Yep. Any more questions? Okay. I'll keep going then. Okay, so we've talked about diagnostics and detection of different diseases. Now now it's important to think about like how can we actually go about treating them with therapeutics. And so this is definitely one of my areas of interest. It's kind of like my area of interest is like once we identify, let's say, a specific cause of a specific disease, how can we make a therapeutic to address that cause? So that's really what my area of interest of research is and where I have a little bit of experience as well. So any questions here that you guys have, feel free to ask as well. But basically, just from a definition standpoint, therapeutic is any treatment designed to help the body recover from an illness, manage some sort of symptom, or stop a disease from progressing. And so it's important to realize that a therapeutic doesn't always have to cure a disease. It's actually really, really rare for a medicine or a drug to come about that actually fully cures a disease. Most of the time, the goal is to reduce the symptoms, to improve a patient's quality of life, or to just slow the disease down so that the body has a better chance to fight it and fight the disease itself and to heal over time. And so um, some of the examples of therapeutics are drugs, which are probably the most common form of it. This can be like a pill, an injection, an ointment, whatever it may be. These are all forms of therapeutics. If you've ever gotten like strep throat or any sort of other, I guess, like bacterial infection, you've probably been administered antibiotics. So that's an example of a drug or a therapeutic that works to kill the bacteria. You think of something like chemotherapy for cancer. If a patient has cancer, a lot of times they're prescribed chemotherapy, which is basically high dose radiation. And I guess like fun fact, not so fun, but fun fact is chemotherapy targets any fast growing cells. And so cancer are cells that grow quickly, uncontrollably and multiply really, really fast, which is why chemo does a good job of targeting them because chemo targets fast growing cells. However, another set of cells in the body that are super fast growing are hair cells or cells that grow at the top of your head, which is why a lot of times you see chemo having that side effect of people and causing them to lose their hair. That's, I guess, a little fact you can take home from today. But That's another example of like a drug. You can also look at something called a gene therapy. For example, I talked a little bit about CRISPR. That's an example of a gene therapy. So anything that fixes faulty genes or introduces new genes or deletes old genes all fall under the umbrella of gene therapy. Engineered cells are super, super interesting in general. And to me, for example, there's different therapies that are out there. For example, one of them is called CAR T therapy, which basically you extract a patient's own immune cells from them, engineer them to recognize a specific form of cancer, and then put them back in the patient. And we've actually seen that that allows the patient's own cells that have been put back into the patient to actually fight the cancer a lot better than it would be possible to otherwise. Um, Another example of a therapeutic, whether or not it's commonly you would commonly think of it, are implants. So whether it's like prosthetics, pacemakers, so like artificial joints, all of these things can fall under therapeutics because they help improve a patient's quality of life. And so the key goals of a good therapeutic are specificity, meaning that it only targets the harmful cells or the affected tissue with minimal side effects or minimal other cells that are affected. So in the example of chemotherapy, again, it's not very specific because it also targets things like hair. However, it is good at its job, and hair is something that a lot of people are willing to sacrifice for their cancer to, get a little better. So that's okay. Again, minimal side effects, and long-term relief or cure is one of the most important aspects of it. You want a therapeutic that'll actually do what it's meant to do. So just going to go through a few different tools, I guess, in the area of therapeutic development. One of them being nanoparticles. Basically, nanoparticles are super, super small particles that we as scientists have been able to use as delivery systems to carry different drugs or therapies into the body exactly where they're needed. And so one of the things that a lot of people in biotech think about, and that's super important to think about, is you can design the most amazing drug known to mankind. If you don't have a way to get it into the body where it needs to go, that's basically useless. Um, for example, antibiotics are great because they do what they're meant to do, and you can take them really easily, like orally via the mouth, and they get where they need to go, which is just the bloodstream because they just need to address a large a large bacterial infection. Um, so it's important to get things where they need to go. If you have something that should target only a drug that should only target heart cells, for example, you really don't want that to be just like floating around in the blood and being able to access all the different cells in the body because that's not what it's supposed to be doing. That's not where it's supposed to be. So nanoparticles allow us, nanoparticles can be designed to basically hone in on specific cells or tissues like cancer cells or like lung cells or lung tissue and actually basically drop the drug off there. And so these are super, super useful, super, super used nowadays in science. Some real world examples of this are um, nanoparticles that can basically deliver chemotherapy drugs to specific forms of cancer. So that's an example. And so nanoparticles are really part of a bigger trend in biotech towards precision medicine. So treating the right person with the right drug at the right time in the right place. That's something that's really, really important in biotech nowadays. Another example of like a therapeutic delivery mechanism is a hydrogel, which are basically like if you've ever seen or like played with like slime, it's basically like that kind of consistency and that kind of material that can be placed basically directly on skin or on a wound. And basically it's made up mostly of water and some sort of polymer. And this the key idea of this system is that it can be loaded with drugs that can be released over time, basically through the gel. And so it can slowly release drugs. It can at a slow, steady, controlled dose, which can help a lot with healing over time. Another tool is engineered cells, which I talked about a little, but basically these are cells that scientists have genetically modified or engineered or programmed to do specific tasks like treat or manage diseases. And so there's some key applications of this nowadays in science. like fighting disease, specifically like cancer with CAR T therapy, like I mentioned. And so again, I'm gonna kind of reiterate this one specifically because I think that this is an example where it really highlights that biotech is doing really cool things and is basically combining different disciplines and different areas together. So basically, in CAR T therapy, scientists take a patient's own immune cells or T cells and reprogram them in the lab to recognize cancer cells and then put them back into a patient. And these engineered cells can then hunt and kill down cancer cells in the patient's body that the patient's old immune cells would not have been able to recognize. And this has actually been shown to be really, really helpful in different cancers like leukemia and lymphoma, even in cases where other treatments may have failed. Another example of where engineered cells might make a true difference is producing like missing proteins. So, for example, if a patient is does not produce enough insulin, you can try a therapy where you basically take their cells, engineer it by introducing a gene to produce insulin and give it back to the patient. And so this is actually kind of how most commercial insulin is made today. Um, it's not made and then it's not an engineered cell that's put back into a patient, rather cells outside of patients that just produce insulin, which is then extracted, purified, and then just delivered to patients. Nonetheless, it's really interesting to think about. And then I guess like last example here are gene therapy vectors, which basically the goal is to replace or fix a faulty gene inside a person's cells. And so you think about how you might want to deliver something that might do this. A tool that would be used to do this is CRISPR. But then when you think about how you actually want to deliver this kind of gene therapy, there's actually a lot of different delivery vehicles that are being engineered and used to do this. And so yeah, kind of wrapping this up, there's a lot of different use cases for therapeutics, whether it's like chronic long-term diseases, like diabetes, which can be treated through insulin therapy, or glucose monitors, whether it's HIV, which can be treated with like antiviral drugs that have been developed, or asthma. But if you know someone who has an inhaler, that is a medicine inside of the inhaler that, again, is doing its job really specifically because it's going straight to the lungs. That's why they breathe it in. So biotech is making all of these treatments possible by developing longer lasting, more targeting, and even things that nowadays cure disease themselves. There's a lot of use cases for new therapeutics in rare genetic conditions, which basically fixing the genes at the source. So if there's a really rare genetic mutation that leads to a specific disease, we can think about what kind of tools we might want to use to actually edit or fix the gene and then actually introduce that system to a person. Yeah, so I guess that is kind of the broad content for today. Again, the point of today is not really to like teach any one topic super, super in depth or specifically, rather just kind of give you an idea of like the broad things that are being done in the field of biotech and bioengineering. So I'll pause here and ask if anyone has any questions about any of the content I've covered, or at this time, I'll open it up to any questions about like anything. If you have any questions about like any of my experiences in college or my research or anything like that, feel free to ask. And then we'll go into like a quick discussion thing in a few minutes just to wrap up if no one has any questions, but would love to answer any questions about anything or like any advice that any of you might want or anything like that. No questions. Okay. In that case, let's go into this. And I would really love it if we could all do this. And then maybe talk a little bit about what we are thinking of or like considering for this. So I guess activity for this week or like right now is like think about one of the tools we covered, whether it's like vaccines, whether it's like machine learning for diagnostics, whatever it may be that you're excited to explore. Um, and then write down one problem area that you're passionate about. So whether that's like cancer or like climate change, pollution, food safety, um, Parkinson's, Alzheimer's, whatever it may be, a problem area you're interested in, try to think of the intersection between the tool and the problem to come up with potential like project ideas or things you might want to look into or things you might want to explore. Um, so let's do this for the next few minutes. Um, try to think of these. And please ask me any questions on any part of this that you might have.
Or should we like write it down and send it in the chat or just think of something?
Sure, you can write it down and send it in the chat. And then yeah, how about we do that and then I'll go through and then we can go through those.
Oh wait, what would you think would be the best complexity level when you're like generating a project on the OpenAdmit thing?
Um, on the platform itself, I'm not too sure, but I think that when you're thinking of projects, it's good to start broad and then kind of narrow your way in as you go. Also, the reason for that is if you, as far as I understand, when you're going to think of a project and it's going to break it down into specific milestones for you. If you think of something that's a little broader, it's easier to think of different milestones that'll actually like get you towards that. So I think that that's really why this exercise is super helpful just because it helps you think of like what tool you might want to use and what problem you might want to solve. And then you can work towards like different milestones. So I think that generally I would say think pretty broadly.
Also, will we be share these slides?
Yes, I think so.
Okay.
Yeah. Okay, I'm getting some answers in the chat, which is good. Wait until we have a few more and then maybe think about some of them and go through them together real quick. Okay. I think most of you sent something. Okay. Let's just go through some of them. And then I guess I might ask the person who wrote the actually I'll ask the person that like wrote the comment to maybe talk about it for like 5-10 seconds, just what you are thinking of looking at, just so we can go in this order, if that's chill with everyone. I guess we'll start with so basically just like say what you're interested in, what you're thinking about, and maybe we can discuss it just like together a little bit. Maybe we'll start with Jocelyn.
Yeah, so I'm interested in like screening tools for um certain eye diseases. So I'm interested in like ophthalmology, which is like essentially the study of the eye.
Okay.
And I'm like thinking about screening a screening device, like a physical device for diabetic retinopathy. So I'm just like seeing how to do it and yeah.
Okay, cool. So you're interested in a physical device then rather than using using existing data. So you're interested in like making the device.
Yeah, I mean obviously we'll have like software that
Right,
will
right.
like technically include those uh like properties, but yeah.
Okay, interesting. Cool. Yeah, so I guess like suggestion there would be so th that really has like two main parts. One, can you make a device that like can can view the eye or like that can like capture images or whatever of the eye? And then two, can you actually make some sort of like internal model or internal like algorithm to identify potential problems? So I guess the first one is more hardware thing, the second part is more software. So for the more like software side of it, definitely I think one step that you could take towards that is trying to look at like what kind of data is publicly available to start thinking about like how you could make an algorithm or model to try to like help you determine whether there is or is not a problem, if that
Got
makes
it.
sense.
Hmm. Thank you.
Yep, cool. And then Annika is next.
I'm interested in like dental diseases and and how I could use these tools to figure out to detect dental cavities and other like causes of these issues, I guess.
Okay, so yeah, so I guess that you would really be wanting to look at then like X-ray scan or X-ray data um of, you know, different like basically just like oral X-ray data. And I think that it's very doable to like train some sort of model or like do some sort of identification of like cavities on those like X-rays or whatever. So I think that that would be pretty interesting as well for sure. Next up, Nalby.
So I was just thinking of like using machine learning to like maybe detect early forms of oral cancer. And yeah,
Do you know what the data for that might potentially look like?
yeah, I think probably I was thinking maybe like saliva because it's like in the mouth, right? So I think maybe saliva or just like pictures. So like maybe like image classification stuff.
Okay. I think that brings like a really important slash interesting point. So when you guys, if you're looking towards um like trying to identify or diagnose different diseases via the use of like let's say saliva or let's say some sort of like the presence of some sort of molecule or something like that, you're probably going to run into like new forms of data that you haven't seen before. So like some of these are like more readily available forms of data. For example, like if you're doing like an X-ray project, you're going to find X-rays online for sure. However, if you want to look at saliva or something like that, you're going to want to look into like different biomarkers that are present in different diseases. So for example, You might not be able to find specifically for saliva, but like let's say that there is data of the like how much of a specific protein is found in patients with oral cancer versus without oral cancer. That's the kind of data you would want to be looking for, like gene expression data or something like that, which basically like quantifies how much of a certain gene is being expressed in the disease state versus in the normal state. And that's the kind of thing that you could train a machine learning model based off of.
How would I find like those like proteins and like how would I like access some of those like data sets?
Um, well, so it a lot of this is like there's not really like one specific place to like find data, but I mean just like general Google searching I think will get you pretty far. Like yeah, like for example, like you just Google something like that. This paper is from 2024, uh, and prediction of oral cancer biomarkers by like protein data from saliva. You look at data availability. The data presented is here, and then like boom, once this loads, ideally there will be some sort of data that can be found here that you know clearly was useful enough to for them to do this paper off of it. So, I mean, that doesn't eh, like I so basically the point is I don't know. Yeah, see, like, look at this. It's like the levels in different cases. So there's a lot of like data science or like data analysis project or like work that you could do here, followed by machine learning work. So that's, I guess, like the general process that I'd recommend some of you might think about following, um, to try to find data for your project. Does that help?
Yeah.
Okay. Um, okay.
I had a question.
Yeah, what's up?
So whenever I'm trying to find out like research topics and ideas, I see like a lot of the things that I've done. I mean, I'm thinking about have already been done before. Do your
Okay.
research ideas have to be completely original, or can you do like kind of a redo or something similar to an existing project?
I think that you definitely can do something that like has been done before because like the idea again it comes back to like what your goal is, right? So when you guys are, in high school, like young, wanting to explore like a new topic or something like that, um, it's much more beneficial for you just like do a project, right? Just like work your way through a project, even if it's been done before. Like these are the people that you're like quote unquote competing against for a lot of these projects are people that are like PhD students or like grad students at a university, right? So a lot of the projects that you might want to try to do have in theory been done before. And that's okay as long as you're like maybe doing something new or like a new approach. Like for example, I think it was Nobby that I was just talking to about his thing. Like this paper is like new, right? This is like 2020, it was published in 2024. And so like I'm also assuming this data set is like relatively new then. So it might be like maybe possible to like do something slightly different from what they did in the paper or like try to explore the data in a new way or something like that. So even if like the main project idea the main project idea could be like the exact same thing, like, right? Like Nabi's project might be also like predicting oral cancer biomarkers via protein data from saliva, but the process to do that can be very different. And I think that that's something you see in science a lot is like people try to do the same task, but do it in different ways. And that's almost always beneficial to like science as a whole.
Okay, thank you.
Yep. Okay. I'm just gonna, I don't know if we have time to go through like everyone's in the chat, but I'm just gonna read some of them out loud. And then if the person that like wrote it has any questions or like wants me to do like a quick Google search for them or like help them out or anything, just let me know. Um, okay. So the next one was I'm excited to learn more about therapeutics, hydrogel patches, and one problem area I'm passionate about is cancer. Okay, so within so hydrogel patches are a delivery mechanism for a different drug. So if that's what you're interested in, it would be maybe a little hard to actually do that at home, like actually make the hydrogel. But I think that there's a lot of research you can do into existing literature to see what kinds of things have already been done and maybe try to use that to like hypothesize or try to design a new way to do it. Um, Tanvi said, some tools I'm excited about are CRISPR and prosthetics. Some problem areas I'm passionate about are cancer and neurodegenerative diseases. Okay. Um, so I think that definitely some of the like comments that I'm seeing, I think like just generally some of the comments I'm seeing like are good, basically like tools and problems that lend themselves really well together to find that intersection. Some of them are a little probably more unclear. So like for example, like prosthetics and like cancer is an intersection where like that intersection is like maybe a bit unclear. But I think that as we talk more about different topics in biotech and as we keep going deeper, we can try to find maybe where that intersection is exactly. Um, uh, Ane said one tool that I was interested in was AI assisted algorithms to classify illnesses. However, my interest lies in diagnosing respiratory and skin related ailments. Okay, so like for example, respiratory ailments, you can look into it's called spirometry, spirometry. It's basically like breathing data. So you can actually collect data, which looks like this, which actually identifies different things like maximum respiratory level, resting, whatever. So this data is available. So I think that looking into things like that might be helpful or useful to you. Microfluidic devices, drug overdoses. Okay, that's another good one. You can definitely find a lot of data on drugs, drug-related overdoses, and the intersection of how we can possibly diagnose those better. Okay. OCD or bipolar disorder. I'm not sure what tools I can use in this case. I think the biggest tools that you could use in this case are like some of the computational approaches that I think we'll talk about more for sure next time. But yeah, I think this is an area where like being able to predict, OCD or predict bipolar disorder based on different scans or different behavioral data might be very possible with, machine learning or some sort of algorithm.
Um, I'm interested in genetics, but I'm like kind of confused on like what I should develop. Like, do you have any ideas? Like that would be good to start off with and not like too hard.
So do you have a specific problem area that you would be interested in? Like,
Maybe like curing like sicknesses and diseases, maybe cancer, or
okay,
like
so
something like that.
yeah, I think that in terms of like what is most doable for like a small scale project for something like this, like when you're in high school, I think like the probably the best like general area you should be looking into is like computational biology or like bioinformatics, right? There's a lot of different like bioinformatics type things you can do with genetic data. Um, let's say, for example, that's like you trying to look for identify what potential genes can lead to cancer. So let's say that you find a huge data set of like gene expression levels in healthy cells versus cancer cells. And you basically do some sort of like data analysis or like whatever it may be to try to identify which genes are more upregulated in cancer cells versus healthy cells. That could be a small project that you could look into. So I think that in terms of what is most doable for, just like at home, I think you should think along those lines.
Okay.
Yeah.
Thank you.
Yep. Okay. Do we have any more questions about anything?
Yeah, I had one question.
Yeah, what's up?
Like going back to mine, you said something about like maybe like since that's already been like done, like 2024, do you think like how would you think like or what can I do to like maybe make some like improvements or like advancements? Would it be like the machine learning aspect and like maybe getting like higher accuracy or something, or like maybe something else?
Yeah, any of that would be like very, very doable. So, I mean, like the thing with science is like things get redone all the time in different ways. Um, there's different, like you can do it in a lot of different ways. It looks like they just did pretty standard like data science type work. If you can try to incorporate machine learning into it somehow, that would be really interesting because I don't think they did it. A lot of times papers also talk about what might be good potential next work to be done. Like a lot of research papers will have like future work or like suggestions like section. So you can a lot of times the authors themselves will just tell you what they envision the next step being, and you can try to do that. Um, but yeah, odds are like if you just do like a different technique or something, let's say that you find a specific technique on YouTube or something that they didn't do, you can just do that. So it doesn't need to be like a super like novel or unique thing that you're doing. I think just like the act of doing it is really important.
I had another follow-up question for my kind of answer. For when you mentioned spirometry kind of thing, do you recommend me like since you mentioned that and I've seen like online that you could find, diseases for like I mean like graphs for like as what asthma looks like in those kind of charts and stuff. And do you recommend that I create a device and then use that use like maybe a spirometry device and then like build on top of that instead of, creating an algorithm, which would be harder to do?
Well, I think actually making the device is harder than making the algorithm. Um, I think that yeah, because like sitting like being at home and just like wanting to do a computational project, there's a lot of data that's available online, um, which makes doing a project like that much, I guess, more feasible. And so I think that I would actually recommend doing like the algorithm side of that and seeing like, oh, how does this, you know, pattern translate to a disease? Like, can I diagnose a disease based on a faulty pattern? Question mark. I think that would be like an interesting question to look at.
Well, I mean, like what I was thinking was, maybe taking like like maybe an already built spirometry device and seeing online like readily available charts of what a normal breathing like charts look like in a normal regular healthy patient, and then seeing what asthma, something like that, and maybe building an algorithm that can recognize it, but through the device's charts that it can create.
I mean, yeah, that's like doable. That's like an extension, I guess, kind of what I just said. So like building an algorithm to identify it and then actually incorporating that with the physical device. That's a cool project.
Yeah, that's kind of what I was thinking because the data that's available right now is probably like recorded in a way that cannot be done by like a regular patient. Like it's recorded with like really high tech microphones, and that can't be done with like a simple iPhone. So it's what I was thinking, maybe using a device.
Yeah, that makes sense. Okay, any more questions? Uh, okay. If not, we'll wrap it up for today. Um, let me, so here, I can I put my email in the chat if any of you have any questions about anything as you continue through the process. Uh, feel free to let me know, ask me anything. But I think we'll wrap it up there. Next time we'll talk more about, I think, some of the computational things that might be interesting to you guys, some of the tools that you might want to consider using if you're thinking of more of like a digital health or like data science or like machine learning based project. So definitely looking forward to that.